This article review is on the gluten-free, casein-free diet in autism, the results of a preliminary double-blind clinical trial by Elder, Shankar, Schuster, Terapue, Burns, and Sherrill, 2006. So, of course, there are many rumors and reports that a wheat and milk or a gluten and casein-free diet can cure autism. However, there is insufficient empirical data to support this. Kate et al. 2000 utilized self-report measures of behavior in blood samples. Another study in 2003 also measured blood samples and antibody levels. Arnold, Heyman, Mooney, and Kirby in 2003 evaluated amino acid patterns, and Kinsberg, Richlet, Hoyen, and Nodlin in 2002 utilized a randomized single-blind study where the results indicated significant changes in the experimental group in autistic behavior, nonverbal, cognitive level, and motor problems. Of course, there are many other randomized control trials to compare the gluten-free versus the non-gluten-free, casein-free diet on autism symptoms, and lots of these focus on metabolic and biological measures, where more research is obviously needed to be done in the area of behavioral measures and the gluten-free, casein-free diet on autistic symptoms. The purpose of this study was to evaluate the effects of a gluten-free, casein-free diet on the severity of autistic symptoms as measured by the Childhood Autism Rating Scale, the Ecological Communication Orientation Scale, and Direct Behavioral Observation Frequencies. To evaluate the effects of the gluten-free, casein-free diet on urinary peptide levels, and to evaluate the role of parent behavior in therapeutic and placebo effects of a gluten-free, casein-free diet in children with autism. So the methods. There were 15 children included in this study. At intake, all the children were administered the CARS and the AIDR. Parents were then provided appropriate meals and snacks based on child preferences that fit with the gluten-free, casein-free restriction. The dependent variables were the CARS, the UPL, the ECO, and in-home observations. The CARS was taken as a baseline assessment. The UPL, the ECO, and the in-home observation were all taken before the diet, at the end of the first dietary condition, and at the end of the study. The in-home observations were coded for child initiating, child responding, and intelligible words spoken, parent initiating, parent responding, and parent expectant waiting. Here is a little chart of the sequence of dietary intervention and measurement. So there's a group A and a group B, and then you can see with the little X's that the UPL was taken on day 1, week 3, week 9. The CARS, the ECOs, the IHO were taken at week 1, week 6, and week 12. So this study utilized a randomized, double-blind, repeated measures crossover design with a counterbalanced dietary assignment. They analyzed the data in a two-sided, two-sample t-test where the dependent variables were the ECO and the CARS. They used a two-sided, two-sample t-test to determine if two population means are equal. The dependent variable was the value in period 2 minus the value in period 1, and the mean for the A-B ordering was compared to the mean for the B-A ordering. Half the difference in the group means is an unbiased estimate for the main effect of treatment, whether or not there is treatment by period interaction. And the two-sample t-test has an advantage over the one-sample t-test because it ignores order, since treatment by period interaction or carryover effects have a negative impact on the one-sample version because of sample sizes for each order differ and because of large variance. The researchers also employed a missing at random model for 3 of the 15 subjects whose week 12 and week 6 data were incomplete on the major variable. So here is another little table of the means and standard deviations by group on week 6 and week 12, and the difference point, which is week 12 minus week 6. And you can see the two group variables for A and for B. So the results indicate the group analysis indicated no significant difference with CARS, ecos, or behavioral frequency. CARS with a p-value of 0.85, ecos with a p-value of 0.29. There is no statistical difference in group data for the urinary peptide levels of gluten or casein with a p of 0.44 and p of 0.11 respectively, and no statistical significant differences in observed parent behaviors, which means there was no detectable parental behavioral influences and or confounds. So the statistically non-significant findings may have been the effect of a small sample size and or large within group variance, meaning there should be more replications done with a larger and more similar population. Researchers noted that even though group data were non-significant for each of the dependent variables, behavioral and language improvements could be seen in some individual children, meaning that while there might not have been statistical significance, there could have been clinical significance on the individual level. There's also a wide variety of behavioral and developmental levels represented in the population in this study. All right, and last but not least, strengths and weaknesses or pros and cons of this experiment. The first being that the parental implementation of the diet could not be adequately monitored, as the gluten-free, casein-free diet items were all administered in the home. We cannot assume a homogeneity of variance, as this was stated several times throughout the article. A less heterogeneous sample would have been desired. There's a very small sample size of participants included in this study. And because we cannot assume homogeneity of variance, we cannot assume a normal distribution. While we can assume that the means are possibly the same, there could be greater numbers of participants at the extremes of the distribution. However, we can assume independence, as each observation was taken independent of the others. The end.